Morning, everybody. Thank you, Rushing and Tom, for hosting the BioEMTalks and inviting me to discuss FibSem for Cryo-ET. So I'm Duncan. I run the Beckman Center for Cryo-EM at Johns Hopkins School of Medicine. I want to say during this talk, then please just interrupt if anything's unclear. So I'm going to be talking about FibSem for Cryo-ET and not so much about Cryo-ET itself. Some capabilities of the Aqualos 2, which you can see on the right. This is our Aqualos 2. CryoFibSem, we use it for lamella generation and sometimes serial block phase stuff, but mostly lamella generation for Cryo-ET. As I said, I'm Duncan, and the work I'm going to be presenting in the second half is done by Dr. Dazong Ding and Kai Kai. So they're my staff members of the facility. The timing is a little bit uncertain, so there should be plenty of time for people to ask questions, even during the talk. And if things are unclear, please interrupt. As I said, I'll be going over two parts. The first part is going to be sort of generally, what are we doing with cryofib? And what are we using it for Cryo-ET, which is basically generation of lamella, cryo-lamella. And the second part will be a new method we're working on here, which is a more rapid lamella lift-out method. That one will be an overview, well, not even an overview, the technical details of how we go about doing that. And if any of you're interested in that and have questions, feel free to enter there too. As I said, I'm not going to be talking about Cryo-ET itself and tomography as such, but that is the goal in the end for making a lot of these lamella. So I want to start at the beginning because I think this is probably the most important part of making lamella for Cryo-ET, which is what are we going to be looking at and what are our targets? So I generated just a quick sort of realistic cartoony thing using ChatGPT, but you have a number of different target samples. You've got neurons or muscles or plant tissues. Maybe you have yeast cells. Maybe you have some sort of soft material, batteries, paints, things like this. You may have nanoparticles. They could be lipid nanoparticles. They could be any number of soft materials. And the thing that's common about these materials is that they're all soft. In other words, if you put them under the standard materials microscope, you're going to blow them completely apart immediately. So we have to do it in cryo, which is why we're doing CryoFibSem. The research targets that we're interested in are generally going to be cells, tissues, or some sort of material. We might be looking at viruses, bacteria, maybe subcellular complexes. Often we're looking at target presence. So did my nano, whatever it is, end up in a cell? And where is it? Once we know what we are trying to look at, we should really consider our research goal. So what is our goal? Is our goal a sub-volume 3D structure? That is the very sexy thing that everybody likes to talk about and has really nice data and really gives you insight into, for instance, what is the ribosome actually doing in the cell without being disturbed or fixed or otherwise changed. However, I think the much bigger number of questions you can ask and that people are interested in is the cellular structural organization. And you can really get quite a good understanding of what's going on in the live native cell or material without having to fix it in plastic and bed it using the CryoFibSem. You may be looking at a moderate resolution sort of structure. So maybe you want to look at the actin network or the structures inside a muscle or something like that. You might not be looking for a 2-angstrom structure. You might be looking for just a 10-angstrom structure. And even then, you can get a decent idea of where the alpha helices are and start fitting in high resolution structures into these things in the cells or even in your nanoparticle or things like this. We may also want to be looking at material identification and characterization in its native state. So down here, I just have the standard workflow for subvolume 3D structure analysis. So again, this is just a cartoon generated with AI, except for the nice picture of ribosomes in yeast that David had made that I put in here. Basically, you window out individual ribosome volumes after you have a nice tomogram. You extract the volumes, you align them in 3D, kind of like single particle, but now we're in 3D. And we have a bunch of other considerations to worry about, which I won't get into. And then ideally, you end up with a high resolution structure of your complex of interest, usually within a cell, but it could be a material thing if it's all the same stuff. And, that could go to moderately high resolution, certainly 5 angstroms, maybe even down a bit lower, depending on exactly what you have. So that's generally what we're looking at. And as you can see, this is a much larger area of consideration than your standard single particle project. There's just a much larger space of things to work with and questions to ask. So before we jump into actually milling our favorite target, let's talk about what kind of sample preparation we want to consider. So we've got a number of different practical considerations to make here. In the end, what we're going to need is, you can see on the right, this is a picture of a grid in the Aqualos 2 in cryo. It's a copper grid, it could be gold, but basically you have some sort of mesh and a thin film and by thin film, I mean 30 microns or less. This could be plunge frozen, or it could be high pressure frozen. It could be a high pressure frozen tissue, anything you want, so long as you can get it thin, but it has to be on the grid. And this is the difficult part often. So you're gonna have to ask yourself, do you have a solution and sample, or a sample and solution, a tissue, some sort of material sample, and how are you going to get it onto the grid intact without it being damaged in some fashion by the freezing process or the processing before the freezing, because maybe you have a bulk material or something and it's oxygen sensitive, or you have a cell that likes to die immediately as soon as you look at it and it moves. But once you have a feeling for that, you need to think about the target throughput required for your research goal. So many questions are sort of a yes, no sort of question. Is something there? Does it look like this? Is it damaged? That sort of thing. And it's sort of a moderate resolution question. Or maybe you want to look at the macro organization of your tissue of interest or complex or cellular thing or material. There's lots of these sorts of questions. Maybe you only need a few lamella. Certainly more than 2 just for repeatability, but less than a dozen, let's say. And then you have the subvolume projects. These are the ones that are I'm going to average a whole bunch of stuff. I'm going to need to average a whole bunch of stuff because I'm just not going to get the resolution that I want without averaging a whole bunch of stuff. This is going to require many lamella, often 100 or more. You may also have a low frequency thing. You're just screening. You want to know, can you find something in your sample? And you may need 100 to go through to see, like, okay, it's only 1%. 1% still a lot, but I need to look a whole bunch because maybe its population is not great in the material. Another consideration is for cells. So if you're looking at a cell, how big is this cell? Are we talking about a neuron that we're growing on a grid? What kind of substrate does it require? If it's something like a neuron and you've got active firing and things like this, once you've grown it on a gold grid, not copper here, obviously, but if I move it out of its currently happy state, is it going to immediately die? This can be a problem and will inform your choice of exactly how you're going to freeze it. Special requirements like gases or just shock stability will be important. If it's a tissue, let's say you want to look at say mouse muscle or something like this. Is it going to survive both the freezing process and the pre-processing process? So how am I going to get a millimeter cube of tissue or material into, say, for instance, a high pressure freezer when I need to cut it down to 20 to 30 microns. Now that's not too thick, I mean, and not too thin, so it's not a huge problem, but it's not also not super easy. Um, the next part is sample preparation complexity. So how much complexity do I have to go through before I can actually get it into my freezing situation of choice? Again, thickness considerations are important. So if I have something in solution, that's not as big a problem unless my cell is huge, and then that can become a problem. And what equipment, this is often a big thing, what equipment do I have available? So I mean, there's very few facilities with every piece of technical equipment that you could have. So, and then the last one that's important is target concentration. So whether that's the cells or the cells of interest in your solution, or just the concentration of the thing you're looking at in the tissue or in the cell. So for instance, for subvolume averaging, if you've got ribosomes in there everywhere, that's fine. If you only have some complex in there and you can only find, say, 10 of them per tomogram and maybe 10 tomograms in a lamella, well, that's only 100 particles. That's just not enough. So this is a huge consideration on whether or not something is feasible. So now that we have some idea about what we are looking at and its requirements, we can talk about whether or not we want to plunge freeze or use high pressure freezing. I did not mention here that you do have cryosectioning. I'm not going to get into that. It's not as explored because we're making lamella here in combination with this. So just to chat briefly about plunge freezing with say a Vitrobot, which is what I'm showing on the right. But you could use a Leica or your favorite setup. This is actually quite difficult for many samples in my experience. So if you're not doing cells like yeast or neurons on a grid, and you're working with a tissue or a material, or something that's more in solution but likes to blot away and not adhere to the grid itself, it can be problematic. It is, however, very good for making thin samples. So you're not going to run into that 20 micron, 30 micron thickness limit, except for, really big cells. So it's quite good for that. On the other hand, I'm showing on the right our equipment, which is a Leica EM Ice, but there's other high pressure freezers out there. You can take your pick. This one's quite nice for what we're doing. It also has light stimulation and various other nice bells and whistles, although we don't use them as much. This one's much more flexible. So there's what's called the waffle method, which I will get into in a bit here. And basically, so long as you can get a section down to, say, 30 microns thickness, you can high pressure freeze it and then make lamella of the targets. So that's incredibly flexible. If it's thicker and it's solid, that is a bit of a problem, which you're going to have to figure out. Maybe you can use a vibrotome or something like that to get it down in thickness. This can be a problem. And I think it is currently really the only practical method for many tissues and materials. So let me see here. So much of this, because we want to go into cryo, we're not freeze substituting, we're not plastic embedding, it's not stable at room temperature. You usually are keeping it at like 4 degrees to keep it alive and happy. And a lot of how to get bulk tissue down to the right thickness is sort of work in progress. If other people have more knowledge about great ways of doing this for sensitive samples, I'd love to hear about them. But certainly here, we're still working on various methods for making that happen nicely. So now here is the main part of what our options are once we have a sample. So I'm going to start this video and just give you a feeling for what does fib milling look like. And we can thank Thermo Fisher for providing this lovely graphic for me, which I certainly am not capable of making this sort of nice graphic. So here we've got a grid in here. You image with the electron beam, and you mill with the fib. You're going to cut away the bulk. This is just a cell on a grid, for instance, a like 400 mesh cryo grid. So mill away the top and the bottom, leave you with a nice lamella that you can use for cryo-ET and tomography. So this is what we're attempting to use or produce. We want a nice thin lamella, something like 100 nanometers. However, often it's difficult to get that, and you're going to be at 150 or 200 nanometers, or sometimes even 300 if the sample is particularly challenging. So this is the normal FibSem lamella preparation that I've just showed you here in this video. There is a few other methods. Let me just pause that. So there's what's called the waffle method, and this is because you're going to take a high pressure frozen sample of cells or your material, whatever it is, and you're going to do an initial scan to find all the grid bars. It's going to look like a waffle. But basically, you're going to do the same thing that you have here, except you're going to do it through the bulk of a, say, 20 micron thick layer of ice and cell and material. Otherwise, it's largely the same. Another method you could try is to lift out your sample. So you could cut out a block from the ice and then stick it onto another grid and thin it down to a lamella of the size that you want while there. This has been done, especially in materials, for a long time. There's a relatively new method in the last, 2 or 3 years was developed called the serial lift out method for FibSem. And this is where instead of lifting out a smaller block, an individual block, you lift out a giant block of, say, a large cell. And then what you do is you kind of slice it up like a loaf of bread, and you lay out all the individual series of blocks. Now, this is the interesting method because what you can do there is get a lot more throughput than you normally would, with the caveat that you've lifted out a single block from a single area. So if you want to look at multiple areas at once, that's more problematic. But a very useful higher throughput technique. And then there's IFLM. So you may be wondering, like, how do I find what I'm interested in? Well, we're going to use light microscopy. In the Aquilus, it's called the IFLM. There is a system called Meteor, but basically you don't look at CLEM images. So I'll just chat briefly about the IFLM design because it will be important for discussing the method and the reason for using the methods for rapid lift out. Basically, you've got a working distance of about 1.3 millimeters. Gives you a lot of flexibility for tilting and moving your grid around to shape it. Resolution is a problem with light microscopy because we're limited by light wavelength outside of super resolution, other correlative techniques and whatnot. But basically, we're sitting in the 300 to 600 nanometer resolution range. In Z, for IFLM at least, we're talking like 2,000 nanometers. Now, if you recall, I want my sample, my lamella, to mill down to, say, 100 or 200 nanometers. So if my target is only 200 nanometers thick, well, this level of resolution is difficult, especially in Z. However, if I can target in XY instead, I've got a much better chance of at least clipping my my sample and getting it in the lamella. So this is just the standard IFLM workflow. So you start out with a nice light microscopy image. with your target of interest. You pick out, part of a cell. Then you mill it down to a chunk. You get rid of the bulk on the top and the bulk on the bottom. Make sure your stuff is still there. And then you thin down the lamella itself, and then again check to see whether or not your stuff is still there. So now I've gone over sort of what we're doing. This is all again to produce a nice thin lamella so that we can do cryo-electron tomography on it. We're going to generate a 3D volume of whatever's in here. And whether or not you want to look at a material with this, or you want to look at, ribosome structures, the whole purpose was to get a nice natively preserved sample. Now, each of the methods that I talked about previously has some caveats about using it. And one of the methods we're developing is, we're referring to it as a more rapid lift-out method. The reason why we're working on this method is because if we want to lift out individual things, it's quite slow. So this rapid lift-out method is good for both high-pressure frozen samples and for plunge frozen samples. Although generally you're going to use high-pressure frozen samples because it's going to be something difficult like a tissue, that would be high-pressure frozen, or material that's sensitive. We're going to actually target things at 90 degrees to the plane of the grid. So that's going to mean that our resolution is better than the targeting in partially Z, which is going to give us a better chance of actually hitting the target because our lamella is actually going to be straight down through the sample. We're going to lift it out as opposed to having it close to the plane of the sample. You can use multiple sample locations. So you can, go around the grid, take it from different cells or material locations or tissue locations. Unlike the serial block method. And it's higher throughput than the regular lift-out method. So at this point, I just want to pause real quick, and if anybody has questions, let me know, and I can go back over, because it's important that everybody's sort of on the same page at this point of why we're making these lamella and what the options are. For some reason, I don't see the chat, but all right. I'll let Tom or Rushing interrupt me if needed. So we're going to start off with the grid transfer setup. So this is the Aquilo shuttle. Inside the Aquilo is just a NavCam image. We are going to have the sample grid, which is our target right here, which is either going to be a high-pressure frozen or plunge frozen sample. And then we're going to have our target grid that we're going to move our lamella onto. In this case, I'm using one of those materials finger grids because I think it's more flexible. However, you could use any number of types of grids. Higher mesh size is probably not great because you limit the areas you can put things in your tilt range in say the Titan Cryos. But you could use a number of different things, and that's fine. One of the first things we're going to do is build or attach what's called a copper adapter block. This method is, as far as I can determine, out of the serial lift-out paper, where they use a copper block, they attach it to the end of our lift-out probe here by welding it on. So in the back here, you can see that we've milled out basically a square, like a bit less than 5 micron cube of copper, and it's only attached by these thin parts onto either side of it. And we're going to weld this to our probe. Welding in cryo SEM is actually pretty straightforward. You mill little slots next to the gap when they're touching. And because it's a cryo and because things like to stick, it's basically all that material is going to float around in vacuum and then re-adhere to stuff nearby and basically weld it to whatever's nearby. So we're going to go ahead and do that. And this is what it looks like in the end. You can see the material built up here and it's nicely attached. So now we're going to just cut the 2 attachment points and pull this out. And let's see, I still don't see the timer. All right, Rushing, how much time do I have?
You're good. You got 3 or 4 minutes.
How many?
5 minutes for Q&A basically now.
Okay, great. Okay, so now that I've attached my copper block, I'm going to cut it out and then lift it out. And I'm going to use this later on. This can be done a day ahead of time, so you don't need to do this the day of. The next part is going to be the lamella preparation. And this is different from the regular lamella preparation in a couple important ways. We're at 90 degrees to the plane of the grid. This is actually a thicker sample, so 20 microns thick. We've milled away the bulk from the top and the bottom to, of a decent distance. You'll notice that the lamella doesn't transit the entire length of the thing that we're going to lift out. We've got a larger section here that we can attach to. We've also got a stress relief and detachment bracket here to make sure it doesn't float away or have other problems during the lift out. So next, we're going to thin the lamella. This is what it looks like once we've thinned it. You could make this bigger or smaller, actually, the lamella itself, but this is practical for what we did. It's about 20 microns, all told. And then this is just another view from the side. David got a very nice picture here of what the lamella looks like that we're going to lift out. So the next part is lamella welding. We're just going to weld it to this copper block, and it works in the same exact way. We turned around because of the way the probes work. And once it's welded, you're just going to cut it loose and lift it out. This is actually, the Oculus is quite nice in terms of its precision, and it's not like it whacks something, typically speaking. So lift out is not actually that bad, even though this does look like a fraught project. It's actually quite stable. We're very happy with it. That's sort of what it looks like on the side. The next part is we need to attach it to the finger or grid of your choice. One obvious way to do it that we first tried is just attaching the bulk portion to the side. But really what I want to do so I can get a nice tomogram is attach it in this manner so that you can get a full range of tilt. And it is nicely orthogonal to the direction of the beam. So you don't have the problem of missing some angular flexibility in the microscope once you get to the Krios. Now, you could also do this on, say, a 100 by 400 mesh and just span 2 sides if you're worried about how much motion you're going to get because it's hanging loose. But we didn't have any problem with that, actually, when imaging it. Again, here's just attachment alternative. Here's the IFL of the lamella to confirm that your target is still there. And that's the rapid lift out method. Now, the reason we're pursuing this method is because you can get up to, say, 6, at least in our hands, although we haven't done it enough times to be really proficient, in a day, whereas lift out is more fraught and a lot less rapid than that. And 6, I feel like, is enough to be a reasonable thing for a low sample copy method to get what you need in a reasonable amount of time. Whereas if you're just lifting out one at a time, you could be talking about a full week of trying to use the Oculus to get to your target. And this one, you can probably get it done in a day or 2. It would be difficult to do sub-volume averaging unless you have a really dense targets sample, or you're willing to spend a week, but still it's possible. I do want to thank everybody. So again, both David and Kai were the people that did the work in the lab for this. And also I want to thank our funding sources, which are mainly Johns Hopkins School of Medicine, and also the Arnold and Mabel Beckman Foundation, which bought a good chunk of our equipment, or paid for at least, helped pay for. and then also the Howard Hughes Medical Institute, who has contributed to some of our equipment as well. As one quick bonus, some of you may have noticed that you get ice contamination everywhere, and it's such a pain, such a pain. And it can look like this. You got just cube ice from, transfer in liquid nitrogen. However, we have a potential solution, which is to clean it. So if you run your ion beam just over the top of your contaminated sample, you will charge it, and it'll all just fly away. So we were thinking about this one of those days. And this is a particularly useful trick because I got to tell you, stuff loves to stick to frozen samples, especially if you've done anything to it with an electron beam or an ion beam. But if you run your ion beam over the top, you can get rid of a lot of that stuff and then be in better shape for your lamella preparation. So with that, I'd like to turn it over and take any questions if there's still time.
Any questions? Nick, are you planning to ask questions?
Oh, yes. Yeah.
Go ahead.
Yeah. Hi. Hi, Duncan. Very nice presentation. And we have Nick at Yale. We also have Accurus too at the core facility. I know it's difficult to provide service for this, especially lift out for lamella. So the two questions for you. The number one question is that have you started to provide service for easy lift out? Not they don't call it easy anymore. They call it lamella lift out. So I know it's very challenging, but if at the end the lamella won't be able to sit on grid well, how do you charge them? The second question is we all know there's a major challenging of ice contamination after lamella prep for either the milling or lamella lift out. So what measure have you been doing to minimize ice contamination before going to cryo-ET microscope for data acquisition?
Yeah, so I'm clear on the second question, which is how do we manage ice contamination, which is a huge problem. So I'll answer that one first. Basically, you use fresh nitrogen. You should have your 4-liter dewars, and every chunk of thing that you're messing with, anything that's going to be anywhere near that lamella, it's got to be fresh nitrogen out of the tank in a dry dewar. Even your transfer station for the Aquilose too, it needs to be freshly dried. You can't reuse it. If you make lamella and then quickly want to take it out, you should probably dry it first. The problem here is the grid cleaning I can help you with the ion trick right here that we used because you got a vacuum in there. Things are not governed by gravity anymore. They're governed more by electrostatic charge. And if you just charge it, which is usually a giant problem in SEM, the charging problem, they'll just fly off. So you can get rid of the stuff that's sort of sitting around because it will go and find your lamella because when you're making a lamella, it's charged. Just like if you glow discharge something, probably even worse because there's no oxygen or atmosphere to do chemical reactions and sort of like get rid of those radicals and like highly charged things that you've produced. So anything nearby that's not attached to something is going to fly right toward it and then like to stick. And the best thing to do is to not have anything that's loose and available for that to happen. So clean nitrogen. If you can manage it, don't leave your lamella sitting in cryo storage. Stick it directly on a Krios or your other system of interest. This goes for materials too. If it needs to be cryo, because maybe you're working with lithium and it's oxygen sensitive or whatever it happens to be. So that's my recommendation for that. We've been trying to think about various different things you could do. You can't really coat it afterwards because that just interferes with your electron beam. I haven't thought of a good way to discharge all those ions, but maybe if there's a atmospheric, and I don't know if we have a thermo person here, but some way to flow like a nitrogen, oxygen gas over the lamella over time, you could let some of those radicals and stuff on the surface react, and maybe they won't be as charged and attractive anymore. These are all just thoughts I had. But can you remind me what the first question was again?
Yeah, first question is regarding service for lamella lift out
Yeah.
service. So what practically the yield rate, the throughput, and if at the end you couldn't get it, it's 1 day work. So how do you charge people as a facility?
Um, so right now we basically just charge. We charge in a couple different ways. So for training, what we'd like to do, because we don't have the staff, especially now, because the administration in the United States is hostile to say visitors and stuff. So staffing's an issue in our case. So we're down a staff member. Unfortunately, David has gone back and found a different job where it's a little bit more less fraught, let's say. But we will basically have a dedicated staff and we are going to be hiring soon again, a dedicated cryo-ET staff person. And I think that's you really need that. You need a dedicated staff person. I think the university needs to support that in the same way they would a facility manager, because getting these projects off the ground is the major bottleneck. Because at least in our case over here, I've got, maybe a doctor that wants to look at some type of tissue for some type of potential solution or something like that. And they've got no idea. And they're going to need to be trained. And they're going to be, to have their hand held and brought from ground zero on a fresh project that somebody's really going to have to think about. And the way we charge for that is basically service covers like sort of their time. So we could charge for that. But really, if they're working with us, it's training, which is heavily subsidized. And if you kind of know what you're doing, but you need us around to answer questions and whatnot, because something comes up, that's assisted use. And that's also even more heavily subsidized. And then if you're just doing everything by yourself, that's regular usage. And at Hopkins, at least, we cover about half of the cost of the facility for that, to bring it into range of what can be afforded with, various grants like NIHR ones. I think there needs to be understanding. You've got a certain number of grants and money out there, and you can charge a certain amount to those. And if you go above that, you're not going to get any more money. You're just going to get less work done and less money. So there's an understanding at Hopkins for that. And so we may be charging for training and assisted use while working out these things, but we charge for that. Sure, it doesn't always work, but I'm very straightforward and clear in the beginning. This is the new project, likely. It hasn't been done before, and it's going to take a lot of work. Even for single particle, this is the case where you're constantly messing with, okay, what are my freezing conditions? Is my sample just junk? You could spend a year, 3 to 5 years working on your sample and screening stuff. We still have to charge for that time when you're screening, even if it doesn't work out. And I use sort of the same approach for lift outs and FibSem stuff, with more warning that these projects are much more individual and varied, and probably you're going to be doing something new. So I hope that helps and
Oh, yeah.
answers the question.